to explain human cognitive distinctiveness, neuroscience often looks to the macro scale, the sheer number of neurons and the complex wiring between brain regions. But the root of our computational capacity must be investigated at the microscale, down at the level of a single dendrite. Over a century ago, Santiago Ramón y Cajal observed that human cortical neurons are uniquely large and highly elaborated compared to those of other mammals. Modern anatomical studies confirm this. Human cortical neurons have vastly larger dendritic trees, elongated branches, and a higher density of specific synaptic receptors than standard rodent models. Yet, knowing these physical differences exist does not automatically tell us how they affect computation. Historically, the field has lacked a rigorous mathematical metric to prove if these extended branches and distinct receptors actually increase the processing power of the individual cell. To understand how our specific cellular architecture shapes cognition, we need a standardized method to quantify the exact input-output complexity of a single biological cell. The solution is to pit the biological cell against a machine learning algorithm. We map the input-output behavior of a detailed compartmental neuron model and force a fixed-capacity artificial neural network to try and replicate it. The benchmark is a strict temporal convolutional network, or TCN, featuring exactly 3 hidden layers with 128 neurons per layer. This gives us a mathematically stable ceiling for computational capacity. First, we bombard the detailed biological model with thousands of random spatiotemporal synaptic inputs across its dendrites. This triggers a distinct pattern of somatic voltage spikes, the cell's natural computational output. Next, the fixed TCN is fed those exact same synaptic inputs. Its task is to predict the biological cell spikes at a strict 1 millisecond resolution. The metric works inversely. If the artificial network accurately predicts the firing pattern, the biological cell is computationally simple. If the artificial network fails, missing spikes or predicting false ones, the biological cell's internal integration is highly complex. This prediction accuracy yields the Functional Complexity Index, or FCI. It converts cellular biophysics into a standardized metric of computational resistance, allowing us to compare the processing power of different neurons directly. This chart compares the Functional Complexity Index across 24 biophysically detailed cortical pyramidal neurons, half from rats, half from humans. The data establishes a clear boundary. The human cortical neurons, marked in green, attain significantly higher baseline FCI scores than their rat equivalents. We can further break down these scores by mapping where these specific cells reside within the 6 layers of the cortex. In the rodent models, functional complexity mathematically peaks deep in the cortex, specifically within layer 5. The human data shows a sharp divergence. Peak computational capacity shifts upward, maximizing in layer 2/3. Mammalian brain evolution involves a reallocation of peak computational power to the upper layers. This directly correlates with the anatomical expansion of layer 2/3 observed in human cortex. To determine the mechanism driving this higher complexity, we must isolate the variables. We need to test if this gap is driven by the physical shape of the cell or by the chemical properties of its receptors. We strip away the human-specific synaptic parameters and run all 24 models using identical rat-type synapses. Even with downgraded synaptic properties, human dendritic geometry alone supports a significantly higher FCI score. As this histogram shows, a regression analysis of 58 distinct morphological features reveals that a single feature, total dendritic area, explains the vast majority of variance. When pairing features, the combination of total dendritic area and the maximum length of bifurcation branches accounts for 81% of the total variance. This physical extension creates severe electrical attenuation. Voltage from a distant branch decays rapidly, forcing dendrites to compartmentalize. Distinct branches become semi-independent computational subunits, requiring local inputs to cross a threshold. The sheer physical geometry of a human neuron shatters the concept of a single-point processor. It forces the cell to operate as a parallel computing array. Geometry establishes the hardware, but synaptic brain chemistry provides the software, specifically the highly voltage-dependent NMDA receptors. Human excitatory synapses contain triple the density of rodents. This accommodates massive NMDA conductance and a steeper voltage dependence, governed by a specialized gamma factor. We test this by driving identical human cell morphologies with 2 different chemical profiles. one using strict rat parameter synapses and the other using human parameter synapses. This graph measures somatic voltage against the number of active synapses. Rat synapses display a slow, linear accumulation of voltage. Human synapses start slow, but right at 35 simultaneous activations hit a sharp superlinear spike. This sudden voltage spike creates a potent nonlinear logic gate inside every isolated dendritic subunit we observed in the morphological data. Human NMDA kinetics convert linear summation into a complex combinatorial logic. The complete functional profile of a human pyramidal neuron relies on the synthesis of these two variables. Extended bifurcation branches divide the cell into isolated physical hardware subunits, and steep NMDA kinetics equip those subunits with aggressive, nonlinear logic gates. Based on the limits of the artificial neural network, stripping away either the expansive human morphology or the dense human receptors forces the functional complexity index to collapse back toward rodent baselines. This framework focuses primarily on NMDA-dependent mechanisms, incorporating active dendritic conductances like sodium and calcium spikes, which currently lack tight experimental constraints in humans, could drive the true FCI even higher. Zooming out to the macro network, the thick, expanded layer 2-3 of the human cortex is populated entirely by these hyper-complex processing nodes. Single neurons operate as deep, highly evolved, nonlinear networks in their own right. The distinct cognitive power of the human brain originates in the architectural engineering of its foundational units.